Okay, we've got the C 13 plus science core biology specimen paper, ISEB, Independent Schools Examination Board. Your time allowed is 40 minutes. So the first section in these ISEB science exams is the multiple choice questions. So we have number one, let's go. Underline the word or phrase which best completes the following. The chemical used to stain the nuclei of cheek cells is glucose, iodine, methylene blue, or water. So we use a stain with cells so that we can highlight the different structures within the cells, the different organelles. Both iodine and methylene blue can be used. However, it's preferable here to use methylene blue. And the reason being, methylene blue binds to DNA. Is due to the electric charge of methylene blue and the negative charge of DNA and RNA. So the methylene actually binds to the DNA and RNA. So it's great for highlighting the nucleus or nuclei inside cells. Okay, let's look at the next question. Osteoporosis causes weak bones. It's caused by a lack of calcium, iron, magnesium, vitamin C. So this question is really asking you, which one of these minerals or vitamins is linked to bone strength? And the answer, of course, I'm sure you got this right, is calcium. Let's read the next question. The structure formed after fertilization is a baby, an egg, a fetus, a sperm. Well, actually, two of these structures are formed after fertilization, but one comes before the other. So the sperm and the egg are the sex cells, and they combine like so. Okay, they're going to join together and they form a structure straight after fertilization. They actually form something called a zygote. This is called a zygote, but the next structure is an embryo, but that's not one of our possible answers. So, and then after the embryo, we get a fetus. So we have an embryo and then we have a fetus. So the answer here is a fetus. Let's go. When people breathe in, air is drawn into their esophagus. Notice the American spelling. In English, we put an O in front of that, but you are allowed to use American spelling. So the air is drawn into their esophagus, intestines, heart, or lungs. And of course, the answer here is lungs. Let's go to the next question. E. In plants, water is transported from the roots to the leaves for photosynthesis, reproduction, movement, respiration. Well, we have to ask ourselves, what is it that the water is going to be used for? And the answer is photosynthesis. The water joins with carbon dioxide to form glucose in a process called photosynthesis. In plants, light is absorbed by carbohydrates, chlorophyll, fats, or protein. And the answer is chlorophyll. Chlorophyll is the green colored pigment that absorbs light. Okay, for our next question, G. The photograph below is Euglena, which lives in freshwater ponds. It is an example of a multicellular organism, an organ, an organelle, or a unicellular organism. It's made up of one cell, so it's not multicellular. It's not an organ. You can't have an organ unless you have lots of cells. And it's not an organelle. Organelles are structures we find within cells. It's a single-celled organism just made of one cell. It's a unicellular organism. When estimating the total number of plants in a field, you should avoid places where there are no plants growing. Place quadrats at regular intervals in the field. Place quadrats randomly in the field. Place quadrats where there are the greatest number of plants. Well, if you think about it, if you have a field and we want to find out, we want to save time. Why do we sample? We sample to save time. No one wants to go around and count 4,000 plants in a field. So if I want to estimate how many are in this field, I certainly don't want to just pick areas where there are no plants growing. or avoid places where there are no plants growing. That wouldn't be very accurate. So what we do is we randomly place the quadrats wherever they may fall. And we do that like 10 times and then calculate the mean. So the answer here is to randomly place quadrats in the field. It's this one here. Okay. We are to identify each of the pictures below as a cell, organ, organ system, or organism. Draw a straight line from each box to the correct box. So, yeah, there we go. The panda, and that's called an organism. An organism is a living thing. The cell, well, we've got a sperm cell there. The brain is an organ. It's made up of lots of cells and lots of tissues doing the same job. And the organ system here is the respiratory system, which is a difficult one because the lungs themselves are an organ on their own. But it does show the lungs, and then it shows some blood vessels around the lungs. But that was quite a tricky one. Okay, let's move on. The picture below shows an insect collecting pollen from a flower. Below is a list of words which identify important parts of a flower. Don't forget to pause the video and have a go at these questions before I go through the answers. Use these words in your answers to the following questions: anther, male nucleus, ovule, pollen, pollen tube, stigma. Name the part of the flower which makes pollen. So which part of a flower makes the pollen? So you do need to know what all the different structures in the flower do. We have a structure in the flower which is called the anther. And the pollen can be orange, it can be red, it can be yellow. There's different color pollen grains in different flowers. Generally, it's orange or yellow. This is called the anther. So that's the correct answer. The insect pollinates a flower. Describe how the insect pollinates a flower. So we want to describe what the insect does. Well, you can see here the insect is landing on a flower and it's trying to get nectar. It wants to eat some sugar, right? It needs to take that back to the hive so it can make lovely honey and other things and feed the queen. While the bee is here, or the insect is here, it gathers pollen from the anthers. They get stuck on it. So pollen from an anther becomes stuck on the bee. The bee then flies on its merry little way and goes to another flower. So here's our bee. Look at the incredible drawing, not. And it goes to another flower. And of course, if by luck and by chance, it may actually transfer the pollen that unwittingly is stuck to its body, it may transfer it to a part of the flower called the stigma, which is normally in the center and hanging out and very sticky to pick up pollen. So when we describe pollination here, an insect pollinates a flower, describe how, we're really describing pollination. So what we want to say is, keep it simple. Pollen is transferred from an anther to another flower's stigma. And the stigma is the female part of the flower that receives the pollen. And essentially, that is pollination. Transfer of pollen from an anther to stigma. Okay, here we are on question 4. People show variation in many characteristics such as eye color, hair color, height, body mass, blood groups. Got 5 there, different characteristics. Pause the video, have a go at the question. A, for characteristics identified above, list those which show continuous variation and those which show discontinuous variation. Now, another name for discontinuous variation is categoric variation. So this means things that change about people or animals or species or characteristics that you can put into categories, distinct categories. Whereas continuous variation is stuff that blends from one to another. Okay, you can't really group it. So let's look at things that are continuous variation that you'd struggle to put in a group. You'd struggle to group people by height. Everybody's height is different. No two people have the same height, etc. You would struggle to put people into groups by their body mass. It just continues. The discontinuous variation, I'm not explaining this very well, the ones that you can put into definite groups, well, you could split the people in your class into people with blue eyes and brown eyes. You could split people in your class with brown hair and blonde hair. So hair color as well can be put in groups. Blood groups as well. You're either group A, group B, group AB, or group O. So blood groups are very definite. There's a definite distinct or discrete, as we sometimes call it, discrete variation. There's a definite difference in categoric or discontinuous variation. So what about eye color? We've done eye color. That's all five. Yep, groovy. We've done them. Let's move on. Okay, bit of data. The height and body mass of a group of 5 boys were recorded. The results are shown below. Jack, Billy, Adam, Aaron, Toby. And then we've got the mean. We've got the height of Jack is 164, and his body mass is 52. Billy is height 152, body mass 49. Adam is height 156, body mass 45 kilograms. Aaron's height is 110 centimeters, and he's 43 kilograms in body mass. And Toby is 100 centimeters and 40 kilograms in body mass. Complete the table by calculating the mean height and mass for these students. Give your answers to 1 decimal place. Now, when they put that, that means if you mess that bit up, you're going to lose a mark. So you need to be able to put these to 1 decimal place. So you whip out your calculator and you want to be typing into your calculator 164 plus 152 plus 156 plus 110 plus 100. You don't get any bonus marks for doing this in your head. And then we divide by 5 and we get the 136.4. So that one naturally comes to 1 decimal place. Let's do the body mass. We have 52 plus 49 plus 45 plus 43 plus 40. And that equals 229. Again, we divide by 5. And this time we get 45.8. Boom. Calculate the range of the height of this group of boys. So you might be wondering, what does range mean? It means what is the range from the smallest, sorry, to the highest. So we know that Toby is the shortest boy with a height of 100 centimeters and Jack is the tallest with 164. So what is the difference? Well, we go 164 minus 100. There is a range of 64 centimeters is the range. You should, in theory, be allowed to put 164 to 100, but there we go. The mean height of 5-year-old boys in London increased by 7.5 centimeters between the years 1900 and 1996. Everybody got a bit taller over this 66-year period. Suggest one reason for this large increase in the mean height of 5-year-old boys in London. So why did people get taller? There are many reasons. The most sensible reason would be improvements. Oopsie, this is no good. Improvements in diet, as height is not just determined by your genetics, but also the quality of your diet. So improvements in the diet or anything that suggests things like that. So more protein, specifically, protein is needed for growth. Anything like that would be suitable. There are some other answers, but they're all rather complex. So question 5, a science class is investigating the behavior of woodlice. The photograph shows woodlice. Woodlice are arthropods. Give 2 characteristics of an arthropod. So if you're a little arthropod, what are 2 of the characteristics? And you need to know the characteristics of arthropods. It's part of the syllabus for the ISEB. So arthropods have no backbone. Do you know what we call creatures or animals with no backbone? We call them invertebrates. So they don't have a spine. They also they have exoskeletons. So exoskeletons is like a shell or a casing on the outside protecting all their soft squidgy bits. So they have exoskeletons. Also, oopsie, they have jointed limbs. So that means their limbs can bend. They have joints in them. Okay, so a few extra bits there. Let's move on. 60 wood lice were put into a choice chamber with 4 different environments: light and dry, dark and dry, light and damp, dark and damp. After 15 minutes, the number of wood lice in each chamber was counted. The class repeated the experiment 4 times. Experiment number 1, 2, 3, and 4. Light and dry, light and damp, dark and dry, dark and damp. Numbers of wood lice in each environment. Calculate the missing totals to complete the table. So for 1 mark, we've got 2 missing totals here. So we just need to, you can see here for light and damp, they've simply added together 4 plus 2 plus 2 equals 8. You can see over here in the dark and damp, 47 plus 49 plus 52, 56, 47, 49, and 52 equals 204. So we can see by comparing to these 2 columns what we should be doing for these 2 spaces here. We should be adding up the number of organisms in each environment and writing the answer. So what is our answer? Well, 1 plus 2 plus 1 is 4. And over here we have 4 plus 8 is 12, plus 7 is 19, plus 5 is 24. So you can work out sometimes in these tables what you have to do by looking at the data that is completely filled in and working out what it is they've done. If you can't work it out yourself, that is. Right, describe the results of the experiment. So now what we need to do is, it's asked us just for a straightforward description of the results. So when you're describing, you don't need to explain, oh, I think woodlice prefer the dark. They love it over there because they can hide from predators. That is an explanation. You don't need to say, oh, woodlice like the wet because they need water, they're living things, and they need to keep hydrated. That's a scientific explanation. That's not what this question is asking you for. This question is asking you for a description. So exactly what you see. So let's look at light and dry. We know that we can look at the light and dry, and we can look at the totals, and we can see that the woodlice prefer the dark and the damp to light and dry. So we can see that straight away. They prefer it dark, they prefer it damp. And then we can get specific. We can compare dry and wet. So we can say woodlice prefer damp to dry. Now, you should really back this up. So woodlice prefer damp to dry. Well, how can we tell that? Well, let's look at these results here. If we look at the dark and damp, there were 204 total in the dark and damp and only 24 in the dark and dry. So they were both dark. So that's not that we're keeping that factor the same, but we've just changed damp and dry here. So we want to focus down on that on our data. So woodlice prefer the damp to dry, as 24 were in dark and dry and 204 were in dark and damp. So what we're doing there is we're using figures to back up our argument. I could have also, to prove that, could have compared the light and dry to light and damp. And you can see again, there's double in the damp here, even when they're both in the light. The only difference is the damp or the dryness. And you can see there, I could have used that data as well, but I've chosen to use that. Right, now we've compared damp to dry. Let's compare light to dark. So I want to do it when the other condition is the same and controlled. So I want to do like light and dry to dark and dry. So we can say woodlice preferred dark than light. What is my evidence? Let's back that up, that they prefer the dark than the light. What is the evidence? Well, only 4 in light and dry, but 24 in dark and dry. So it was important there that we keep the dry the same because we just want to compare the dark to the light. And you can see there's a lot more preferring the dark, even when the conditions are like of dry and dampness are the same. Lovely. And that will get you all of your marks. So let's move on. Suggest why it was helpful to repeat the experiment 4 times. So why do we repeat experiments? More repeats gives us more data. Okay, so when we do more data to compare, we like to compare the data. When we do repeats, we can compare and see if there's anomalous results. We can spot odd results. Another word for odd results is anomalous, like this, anomalous. So we can spot odd results, making a calculated mean more reliable. So, and also we can calculate a mean. If we don't do a repeat, we can't calculate a mean. If we do the repeat 4 times, our mean is going to be more accurate. Let's move on. Okay, we're nearly through here. We're up to question 6. In wine-growing regions, warm temperatures can cause high rates of photosynthesis in the plants that produce grapes, which make wine. Wine contains alcohol. Name one other factor which increases the rate of photosynthesis. So let's scroll down here a little bit, or zoom out a bit, actually, so that we can get all of this in. Okay, name one other factor which increases the rate of photosynthesis. So what do they mean, other factor? They mean other than warm temperatures. So they've already identified for us that warm temperatures. What else could speed up the growth or the food production of plants? So we could increase carbon dioxide. And have a look in the picture. What's the most obvious thing? They're giving you a clue in the picture. What's this? Light. So the 3 limiting factors of photosynthesis are carbon dioxide, light, and temperature. You may think water and number of leaves as well, like the amount of chlorophyll in the leaves. They're also good answers. Water is not normally one of the limiting factors, but technically they should accept water, hence why you water plants. Now let's look at the next question. Suggest why it's an advantage to wine growers to have high rates of photosynthesis in plants which make grapes. So why do wine growers want high rates of photosynthesis? Well, they can plants produce more grapes, and not just more grapes, but faster. So they can produce more wine and make more money from selling the wine. Moving on. Clear that. Alcohol is made during fermentation. B1. Name the organism which is used to make alcohol in fermentation. So that's a type of fungi that we call yeast, and yeast is in the kingdom of fungi. Name the kingdom to which this organism belongs. I kind of gave that away, didn't I? Fungi. Do you know the other kingdoms? You should know all of your kingdoms. You have plants, animals, protists, fungi, and bacteria, or prokaryotes. Okay, let's move on. Complete the word equation for fermentation. There's a big mistake that people do here. They don't read the question properly and they start writing the symbols. It's asking you for words, not the chemical symbols. So we want glucose. When yeast, and you do not need to do this, I'm doing this, but you don't need to write yeast there. But when yeast break down the glucose, what do they make? They make carbon dioxide and they make ethanol. Or alternatively, you could put alcohol. Ethanol is the specific alcohol that they make, and they release some energy. So don't be writing CO2 instead of carbon dioxide. They might not give you the mark because they specifically asked for the word equation. C, explain why it is not a good idea to drink too much alcohol. Well, alcohol can do lots of bad things, but the things they're mainly looking for is liver damage. This is liver damage, brain damage, and help you put on weight. It's a depressant. So it's not good. Apart from not drinking too much alcohol, what 2 other lifestyle choices a person can make in order to be healthy. So you can increase your exercise, so more exercise, diet, lower in salt. I'm just going to list off some of these here. Diet, lower in saturated fats. Now, some fats are really, really good for us. So a diet lower in saturated fats would be good. More exercise, a diet lower in salts, not smoking. Yeah, not smoking tobacco or cigarettes. There's lots and lots of other things we could do. Let's go. Doing the last question. Question 7. The photograph below shows a polar bear in the Arctic region. My gosh, he's running out of ice, this poor kid. Suggests 2 ways in which the polar bear has adapted to living in the Arctic region. All right, how's it adapted to living in the Arctic? So we got any suggestion. It's got colorless fur. We could say white fur for camouflage. Okay, so white fur for camouflage, colorless fur actually allows more sunlight to go through. I didn't really need to put colorless there, but we could, sticking on the fur theme, we could say thick fur. So don't just say it has fur, because all mammals have fur. The polar bear has thicker fur than other mammals. So thick fur, it's got thick fat layer called blubber. And the thick fur and the thick fat layer helps insulate. So that's the explanation as to why it helps. It's got claws for killing prey. Again, we could go down the sharp teeth for killing prey. Well, yeah. It's got, I'm going to run out of room here, but it's got small ears, which reduces heat loss. It's not so that it can't hear. That would be a really rubbish adaptation, so that it can't hear its prey. They don't have predators, but yeah, small ears is to reduce heat loss. It reduces the surface area to volume ratio of the polar bear and therefore reduces heat loss. Cool. It's actually got black skin. A lot of people don't know that. It has colorless fur and black skin, which absorbs heat. It's better at absorbing heat. Okay. White skin would reflect heat and keep it cool. That's probably not a good thing. Okay. So in the summer months, the ice caps are melting at an increasing rate. Polar bears need the ice to enable them to hunt seals. If the ice melts too early in the summer, then hunting becomes more difficult as they have to travel further to hunt. Suggest how melting ice may affect the survival of the polar bears. So the fact there's less ice and they can't eat as many seals, is that going to increase their survival or decrease it? So the first statement is it's going to make surviving harder. So how's it going to affect the survival? Decreases survival, which I can't spell, survival chances. So it actually makes it harder for them to survive or less likely for them to live. So you can put anything along those lines to suggest more polar bears will die. So decreases survival chances as harder to catch prey, which is the seals. So less food, less chance of survival. There you go. They're going to use more energy. There's other answers. You could say that they expend more energy trying to hunt the seals. It tells us they have to go further and hunt further. So they're going to use more energy, which means they need more food to survive. And the very fact they're getting less food makes it all very difficult for the polar bears to survive when there's less ice. Let's zing down and finish up this paper. As well as seals, polar bears will eat other foods such as berries, eggs, plants. Seals eat codfish. Cod eat smaller fish, which feed on microscopic plants. Use this information to draw a food chain for the Arctic. So what we need to do here is identify the producer. So we want to identify the producer. We've got microscopic plants. So we can start over here with microscopic plants. A common mistake is to start with the top predator. You always start your food chain with the producer. And what eats the microscopic plants? We've got the small fish, which it tells us in the writing there. And then what eats the small fish? Well, it tells us cod eat small fish. You can see that right here. Cod eat small fish. The small fish feed on the plants. So we've got the plants, we've got the small fish. Our next one is cod. And then what eats the cod? Let's have a look. if we check up here, it tells us, as well as seals, polar bears will eat other foods such as berries, eggs, plants. Seals eat codfish. So we now know that the polar bears don't eat the fish, or maybe they do. But the question is telling us that the seals eat the fish. And of course, the polar bears eat the seals. So we have a 5-level food chain. 1, 2, 3, 4, 5. Name the source of energy for this food chain. So what we're really looking at is, if we go back to the plants, they're the source of energy for these guys, all of these animals. But what's the source of energy for the plants? Of course, that'll be the sun or sunlight. Okay, let's move on. We're nearly there, guys. Nearly at the end. Changes in sizes of populations of polar bears in the Arctic were estimated in 2005 and again in 2009. Here are the results shown. Changes in the size of polar bear population. 2005, we've got stable, increasing, insufficient data, and declining. In 2009, we've got a declining population, stable, increasing, and insufficient data. Explain how these results support the hypothesis that the population of polar bears is under threat. So what we can see here is that if we compare the stable populations between 2005 and 2009, there is a decrease in stable population and decrease in increasing population. So there has been a decrease in stable populations and increasing populations, which means areas where the polar bears were growing in number have gone down, and areas where the polar bears, their population has stayed stable, has gone down. What do we have an increase in? Look, the areas where there's less and less polar bears, there are more areas in the Arctic which have less polar bears. So what we would say is declining populations are increasing. And that will get us our marks. And the areas for insufficient data is pretty similar. All right, let's move on. Hungry polar bears sometimes visit towns in the Arctic looking for food. They are often hunted and shot dead. Suggest why local residents might hunt and shoot polar bears which visit their towns. Well, obviously, polar bears are dangerous, so they can kill people. They can kill humans, so that's a threat. What else? The polar bears might compete for the food for the town. So it's not just that the polar bears are dangerous and they might kill the humans. The polar bears may steal the food or compete for the food that the townspeople are eating. So if the townspeople are hunting seals, the polar bears may eat the seals, etc. When light shines, question 8, by the way, when light shines onto a piece of Canadian pondweed, bubbles of gas are given off. This is shown in the diagram below, which represents a section through the stem of a piece of Canadian pondweed. So this is a stem, and we can see some bubbles coming out of the stem. The leaves have been removed. These gas bubbles contain a mixture of gases as shown below. Gas, oxygen, carbon dioxide, nitrogen. Bubbles from the Canadian pondweed, 50% oxygen, 6% carbon dioxide, and 44% nitrogen. If we look at the air bubble, a normal air bubble is only 20% oxygen, hardly any carbon dioxide, 0.03, and a lot more nitrogen. Describe and explain the difference in the concentration of oxygen in the bubbles between the Canadian pondweed and the air. Well, what does the pondweed do? It's a plant, and plants, in order to make their own food, do photosynthesis. So the difference is photosynthesis produces oxygen. The pondweed photosynthesizes. Cool. Let's move on. For another 2 marks, describe the evidence which suggests Canadian pondweed is respiring. So what's the evidence that it's actually respiring? Well, if it was just photosynthesizing, it would have even less carbon dioxide than the normal air because photosynthesis uses carbon dioxide, but its carbon dioxide level has gone up. In fact, it's gone up many, many times greater than the normal carbon dioxide level in the air. So it is producing carbon dioxide via respiration. We can see this as the carbon dioxide concentration is 6% compared to air's 0.03%. So you could, if you wanted to be really fancy, you could do some maths here and go 6 divided by 0.03 and say that it's 200 times more. Explain why green plants such as Canadian pondweed are important organisms in river ecosystems. So they produce oxygen, which allows other organisms to respire. So without pondweed producing oxygen, we wouldn't be able to get other creatures living in the water. Also, they are a food source. Remember, they're right at the bottom of most of the food chains for animals. So 2 reasons: they produce oxygen and they're there to be eaten. So super duper. Right, final question. Serena and Tom want to find a best way to keep strawberries fresh, or the best way to keep strawberries fresh. They put the fresh strawberries in 3 containers as shown below. 3 types of container: a plastic box with a lid, a sealed plastic bag, on a tray open to the air. Serena and Tom keep the strawberries in a warm room. They observe the strawberries every day to see when the strawberries become soft and wet, soggy, or covered in white mold caused by a fungus. We call that moldy. Their observations are shown below. Let's zoom in on their observations of the 3 strawberries packets. Let's have a little zoom. Type of package. So when they just had a plastic box and a lid, the number of days until the strawberries became soggy or covered in white mold was 4 days before they got moldy. Soggy and moldy. Sealed in a plastic bag, it took 5 days. They got soggy, but they didn't get moldy. And then when they were just kept in the open air, they went off or bad in 2 days. They got soggy and moldy. Name the independent variable. This is, what are they changing in this experiment? The independent variable is always found in the first column in a table of results. So it's the type of package. Then our next question, write a conclusion from these results. So the conclusion is what we found out. The best container for storing strawberries is the sealed plastic bag, not nag, sealed plastic bag. And we would back that up. It took longer, 5 days before going soggy. And it didn't go moldy. So not only did it take longer, 5 days compared to 2 and 4, if we wanted to include the data for the others, but it didn't go moldy. So the strawberries lasted the longest. We could say we could also put in our conclusion here that the tray open to the air took the least time to go soggy and moldy. And that is the end of the test. How did you do? Mark it out of 60. Hope that was helpful. Like, subscribe. Let's get to the marks.
You're still here? It's over.